Okay, to those of y'all just waking up and joining us, good morning. And to everybody else, good afternoon, good evening, and good night. This is Mr. Aguilar. I'm sure you guys are just buoyantly happy to hear my voice for the first time in a while. It's good to be here. I'm going to be going over some PCR stuff with you guys today. It's a PCR Virtual Lab. It is brought to us by the University of Utah. Learn Genetics. I will make sure you guys have all the link associated in your class Teams pages. Now you guys can go along and do this with me, or you can just pay attention, write some things down, and go back and do it on your own later. I would love for everyone to do it themselves, just to kind of get a feel for things. Now I did also make an interactive study guide to go along with this, so again, if you want to try to follow along with that while I'm working through stuff, that is okay. You may find that things go a little quickly. That's okay too. Again, I'm going to keep this video up. You can watch it as often as you want. You can run through the lab by yourself. Whatever floats your boat, guys. I know these are crazy times for all of us with COVID-19 and the coronavirus, and we're all just kind of learning day by day. But, well, we're going to try something new, and we'll see how it goes. Okay. So I will give you the link to the PCR Virtual Lab through the University of Utah website. It is going to look like this. And where do you think we're going to go first? I think we're going to go to begin. Now, I'm going to go ahead and read the slides. I'll describe them a little, and then we'll run through it. Hopefully, we can get this done in the 15-minute time frame that we have. Okay, so the human genome is made up of about 3 billion chemical base pairs. Now, scientists often need to isolate a very specific segment of DNA from within a vast amount of genetic material. Since this segment is just a tiny piece of the genome, they need to have many copies to work with. Okay, so you're trying to study a specific gene or something else, and there's a small sequence of DNA that codes for it. Anytime you want to study something, it's better to have more of that something than less of that something. Imagine if you were trying to study ants and the behavior of ants and you only had a single ant. How good is that going to do for you? Not all that good. You have a whole colony of ants, all of a sudden now you have something to work with. So PCR is a way to take a specific segment of DNA that codes for a gene or a protein or something else, and we can make many, many billions of copies of it, which is great because the more copies we have, the easier it is to study and manipulate. And as we know, we find our DNA usually in the cell nucleus, as we can see right there. It's a strand of our DNA, multiple strands. So polymerase chain reaction is what we're talking about. That is PCR, which can generate hundreds of billions of identical copies of a specific DNA sequence in a matter of hours. It's crazy, right? As the inventor of PCR explained, the technique requires no more than a test tube, a few simple reagents, and a source of heat. So that's the greatest thing about it. You don't need a whole bunch of crazy material and expensive equipment. A few reagents, a source of heat, and a test tube, and you can do these kind of reactions yourself. Okay, lots of DNA. So we can get DNA in things like a hair follicle, which they're going to show us right here. So to perform a PCR reaction, you need DNA that has been extracted from cells, obviously. Because the purpose of PCR is to make more DNA copies, you don't need a whole lot of DNA to start with, which is wonderful. For example, you could extract DNA from something as small as a sample of blood, skin, saliva, or even hair follicles. So think about like on CSI and stuff with forensic guys go in and they're looking for just even a single hair follicle can be enough DNA to where we can make billions of copies and study it, just like on TV. Okay, so the first thing we're going to do, this is where it gets kind of interactive. We're going to go ahead and take our extracted DNA. So let's say we got a hair follicle, we extracted DNA from it. Now we're going to put it in a special tube. Now the way PCR works is by it heats and cools a solution many, many, many times over. So the PCR tube is going to be designed for even heat distribution. So I'm going to go ahead and take my micropipette right here. I'm going to grab my DNA. I'm going to hold the plunger down. I'm going to keep holding, and then I'm going to extract it into my tube right here. Now that we have the DNA in our tube, we need to add primer to the PCR tube. Now what primers do is they attach to sites on DNA strands at either end of the specific segment you want to copy. So the primers aren't random. They're specific primers to isolate a specific segment of DNA. Now they are very powerful tools for copying very specific sequences of DNA, like I said, and there is almost no chance that they'll ever target the wrong site, so they are wonderfully efficient. Let's go ahead and grab our primer. Again, I'm going to hold down the plunger, keep holding, drag it over, and there we go. Now I'm going to add primer 2. So primer 1 attached to one side of the DNA, primer 2 is going to attach to the other side. So imagine you were trying to edit a video and you need to cut out a specific scene. You need to cut one before the scene and you need to cut one after the scene in order to isolate it. So now we have primer 2 into our tube, holding down the plunger. Okay, next thing we need to do is add nucleotides to the PCR tube. Why do you think we need nucleotides? What are nucleotides? Remember, nucleotides are the building blocks of DNA. So if we're going to build a bunch of DNA, imagine like we had Lego pieces and we wanted to build a big castle. Well, in order to build a castle, even if you have the instructions, you still need the Lego bricks to build the castle. So this is kind of the same thing. Nucleotides are our Lego bricks to build our specific sequence that we need. So we need billions of DNA nucleotides to make billions of DNA copies. Let's grab our nucleotides right here, plunger down into the PCR tube. Alrighty, lastly we need DNA polymerase to be added to the PCR tube. Remember, DNA polymerase acts like a tiny little machine that reads the DNA code and then adds matching nucleotides to create DNA copies. This particular DNA polymerase is able to withstand high heat because remember, our thermocycler, which we're going to get into in a minute, is going to raise the heat by a lot and then lower it in order to do some different things we're going to talk about. So the DNA polymerase, again, that's a little machine that takes all the nucleotides and puts them all together in a row, which we'll see in just a minute using complementary base pairing rules, which hopefully we know a little bit about. We need to grab our DNA polymerase. Uh-oh. Hold the plunger into the tube. Okay. All right, PCR tube is now filled with everything we need, so it's going to go into the thermocycler, aka DNA thermal cycler. This machine can precisely heat and cool your tube at specific times, which will allow for the primers and the DNA and all the other reagents to do what they need to do to produce those copies. So let's see how it works. Go ahead and put our little top on the tube, and I'm going to drag it into the thermocycler. Let's go ahead and hit the start button and see what happens. I'm guessing we're going to heat it up first. What do you guys think? All right, here's our target sequence right here, piece of DNA. So we're going to start cycle 1. Thermal cycler is going to heat up to about 95 degrees Celsius. That's 203 degrees Fahrenheit. If you guys remember, 212 degrees Fahrenheit is boiling, so that's very close to it. So at that temperature, the DNA is going to begin to unravel. So imagine those of you guys out there who might have curly hair and you use a curling iron. Hair is protein. When you apply heat to a protein, it takes those curls and it flattens them out, denatures it. Same thing's going to happen to the DNA. The heat is going to unravel it and pull it apart and make it straight instead of curly, just like a hair straightener. So there we go. We see the heat comes in, DNA gets pulled apart, and then we're going to cool it down. Now, when you cool it down, the DNA itself is going to want to come back together, but it's not going to be able to. Those primers are going to get in the way and they're going to attach and they're going to keep it pulled apart. So as you can see, the thermal cycler is going to cool down to about 50 degrees Celsius, which is 122 Fahrenheit. So it's going to go down about half as much as it was before. DNA will try to pair up, but the primers will basically crowd the DNA out. They will lock in and prevent them from re-annealing or coming back together. Now we have our DNA polymerase. Okay, so our thermal cycler is going to change to about 72 degrees Celsius. That's going to activate our DNA polymerase. The polymerase is going to locate the primers attached to the DNA strands, the single strands, and it will begin to add complementary nucleotides to the strand. So let's remember, A always goes with T, G always goes with C. So as long as you have a single strand of DNA, if there's an A there, it'll pop a T. If there's a G there, it'll pop a C, and it'll just go on down the line to build a whole new segment of the DNA in question. Once it's done, it gets to the end, falls off. There you go. And we see it go, attaches everything it needs, and they fall off. Now that's cycle 1. Let's see what happens in cycle 2. Oh, what's going to happen? What's going to happen? Oh, you know what? The exact same 3 steps. Every cycle is going to be just about the same, making more and more and more of the DNA sequence in question. So temperature is raised, going to separate the DNA strands. Primers are going to attach. The temperature will be lowered. The primers will attach when it's lowered. They will keep them from re-annealing or coming together. There you go. DNA polymerase comes back, pops in all the gaps. So as you can see, temperature is raised again by a little bit to stimulate DNA polymerase to copy the strand. Let's see what happens in cycle 3. Probably a little more of the same if I had to guess. And now we have our desired fragments. During cycle 3, your desired products begin to appear. 2 strands that begin with the primer 1 and end with primer 2. These are DNA copies of just the segment of DNA that you're targeted. So that's just the sequence we wanted. Now, although there are only 2 desired fragments right now, you will continue with the cycles. These products will become essentially the majority of the mixture. So we'll go from having maybe 1 possible DNA sequence to hundreds of billions within a matter of hours. That's crazy, crazy. And we're going to see it going. We're seeing the temperature going up and it's going down. Now, cycle 4, we have 8 fragments that contain only our targeted sequence in question. One more cycle goes through. All the 3 steps happen again. Now we have 22 fragments. So as you can see, we're almost going up by an exponential amount. Every cycle makes more and more than the cycle before it. So let's go ahead and fast forward here a little bit. And boom, boom, whoa. Hopefully nobody has epilepsy because that could give you a seizure. All right, so after about 30 cycles, there are over 1 billion fragments that contain only the target sequence and only 60 copies of the larger length molecules. So that's actually a really efficient method of copying DNA sequences. We now have a solution of nearly pure target sequence. Now that's PCR, guys. So again, you could be saying, okay, so we took a segment of DNA and we made a whole bunch of copies of it. I still don't know why. Once again, the whole reason we're making copies is to study it. Maybe you want to study if a gene is turned on or off, when we were talking about mRNA and such last time. Maybe you want to catch DNA, like I said, you were trying to catch a killer or a thief or someone who left DNA at a crime scene. Well, you need lots of DNA to study it and to compare it to other things so you could get a conviction. So again, DNA has many, many, many different things we can do with it. Studying genes, studying diseases, studying health attributes. When we're going to be talking about genes in space in just a little bit, this is what it's going to be dealing with. We're going to be coming up with science experiments that could be done in space utilizing PCR. So basically studying DNA up in space and using this machine to do so. So really cool stuff, really cutting edge. I know I get a little bit crazy on these things. Hopefully y'all learned something. If you didn't, watch the video again. If you still don't, hit me up. You know where to get at me. Shoot me an email. Get me on the chat. I am going to start having office hours most likely from 1. You guys know you can always contact me anytime. But I'm going to make sure I am sitting at my desk from 1 to 2, probably Tuesday, Wednesday, Thursday. That way, if anyone has any specific questions about things we do, you can talk to me. If we even need to have a Zoom session, not a Zoom session, I'm sorry, a team session, we can do that. Whatever works for you guys. I will also be making an announcement later today concerning some mentoring sessions that Dr. Sweeney is setting up with some med students. Because again, we are going to be doing something called a Genes in Space contest, where we will be thinking up crazy different experiments concerning DNA and the use of PCR up in the International Space Station. This is real stuff. It's a real contest that goes on. BCMA has won certain awards every single year. A couple years ago, we actually got to go to NASA and talk to astronauts using video messaging in space while they were in space. It was crazy. So anyways, guys, I'm going to make a few announcements about that. That'll likely take place from 1 to 2 o'clock, Tuesday, Wednesday, Thursday of this week, where you guys can come in. Now, that's going to use Zoom. It is not going to use Teams, so we will send you a separate invite for that. But you will be able to talk with actual med students and doctors to try and come up with some cool DNA experiments. Anyways, guys, it has been wonderful talking with you. I can't wait to see y'all's faces, hopefully sooner than later sometime. I know HISD is closed, but you know where to find me, guys, and I'll get back with you shortly. All right, everybody, stay safe, do what you got to do, try not to be too bored. If you need anything, you know where to reach me. All righty, bye-bye.